Hello friends, welcome to BMH Learning. This video is about restriction mapping. A restriction map is a description of restriction endonuclease cleavage sites within a piece of DNA. This uses specific restriction enzymes to cut a specific sequence of DNA known as restriction sites. A restriction map shows all the locations of that particular restriction site throughout the genome. For example, the restriction enzyme EcoR1 always cuts at the sequence GAA-TTC/CTT-AAG. A restriction map shows all the locations of that particular restriction site, shown here GAA-TTC throughout the genome. There are two specific types of restriction mapping: optical and fingerprint. These types are discussed in separate videos. One common method for constructing a restriction map involves digesting the unknown DNA sample in three ways. Here, two portions of the DNA sample are individually digested with different restriction enzymes. For example, EcoR1 and BAMH1. And a third portion of the DNA sample is double digested with both restriction enzymes at the same time. Next, each digestion sample is separated using gel electrophoresis, and the sizes of the DNA fragments are recorded. The total length of the fragments in each digestion will be equal. However, because the length of each individual DNA fragment depends upon the positions of its restriction sites, each restriction site can be mapped according to the length of the fragments. This was all. Thanks for watching.